the previous video we have understood that it's the beta 4 region of the Smad3 that plays an important role towards cell motility in the TGF-beta induced cells. So here in this video we will discuss what are the amino acids in the beta 4 region that cause this uh, responsibility, or that plays the role towards this cell motility in the TGF-beta induced cells. So from the heading it's clear that it's the Arginine-104 and 105. So how they come up to these two amino acids? They did the mutagenesis analysis and finally come up to these two amino acids that if are important, and if they are replaced by other amino acids, then this Smad3 will lose its functions to control cell motility of the TGF-beta induced cells. So in the panel A we have seen that the results for this Arginine-104 and this Alanine-105 mutated cells, and we have seen its C-terminal phosphorylation, whether it's get triggered or not. So in the wild type A549 cell lines we can see that phosphorylated Smad3 pathways is there, but for the knockout it's not there. But when they are transduced with the Smad3 we can see the phosphorylations, and for this RK pathways, when the cells are transduced with mutated Smad3 at Arginine-104 or this Arginine-104 and Alanine-105, we can see that phosphorylations again get triggered when their cells are induced with TGF-beta for 1 hour, and this indicates that the Smad3 pathways regain its ability once these mutated arginines are induced. And thereafter they check this luciferase activity, and in this they are able to find out that luciferase activity is also gets enhanced when the cells are injected with TGF-beta. And they did it when the cells are knockout cells, are Smad3 knockout cells are infected with this Smad3 knockout wild type, or this mutated Smad3 at Arginine-104 and Alanine-105. And after that they check this. They check the they check the cell migration capabilities in the panel C, where they can see that the wild type cells has more migrations compare after cells are induced with TGF-beta for 12 hours, but knockout cells does not have too much change in the migrations with and without TGF-beta. But when these Smad3 pathways are infected in these knockout cell lines, they see that migrations is there, or proliferations are also seems to be there. And when they use the mutated Smad3, that is arginine is replaced with K, they are able to see a less migrations. And when both this arginine and alanine are replaced, they are able to see some increase in the migrations compared to the single mutations. And these things they have quantified on the right hand side, where you can see that so when the both are mutated they do not see too much changes, but when single one is mutated they see small changes. These things are quantified on the right hand side, where we can see that in the case of wild type A549 the migrations is more, but for the Smad3 knockout they do not see much migrations. But when the Smad3 is infected, wild type Smad3 is infected, they see the migrations, increase in the migrations, which is comparable to the wild type. And when the arginine is replaced, they see a increase in the migrations, but to a lower level compared to the wild type. And they did it for two different clones and both shows the similar results. But when they did it for the dual mutations, that is Arginine-104 and Alanine-105, they see no increase in the migrations, indicating that this dual knockout amino acids, or the Arginine-104 and Alanine-105, are key regulators on the beta 4 region of the Smad3 for TGF-β induced cells. Thereafter, they did the wound heal assay, where we can see that the wound heal capacity is high for the wild type F549 cells. It goes down for the Smad3 knockout, but when they are infected with this knockout, are infected with this Smad3 wild type, they regain this capability of wound healing. And in the case of dual knockout, they are not able to regain the wound healing capacity. And these things are quantified on the right hand side of the penalty, where we can see that relative distance is high in the wild type cells with TGF-β, but for the Smad3 knockout I do not see an increase. But when the Smad3 knockout are infected with this wild type Smad3, they see a further increase in the relative distance traveled by the cells. And when this dual knockout is there, that is arginine and alanine knockout is there, we do not see increase in the migrations. And in the panel it shows the position of the Arginine-104 and Alanine-105, and they hypothesize that they plays a role towards binding of the uh, proteins to the binding of this Arginine-104 and Arginine-105 plays a role towards binding of the proteins with the other proteins that might trigger these ECM characteristics, that is epithelial to mesochemical characteristics, that will cause more migrations. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. I hope you understood how they finally validate their hypothesis about Arginine-105 and Alanine-104 knockouts plays a role towards this cell motility in the TGF-β induced cells. That's all for this video. Thank you for your time.